Today I'm going to talk about some modeling of long-term survival outcomes in R. This is something that I am Yolanda Hager, and I'm a postdoc in the Applied Math Department at CU Boulder, finishing up there. And I've been working with Vanya Dukic on a few different projects, and one of them is this design and implementation of model that can analyze long-term survival outcomes and the accompanying R package. So just to briefly cover what survival modeling is for people who maybe aren't as familiar with that, survival models are used for trying to characterize time to event distributions. So usually in the simplest case, in the most common case, the event is binary, so we either see it happen before some observation period ends or we don't. Examples of this, the most classic example would be time from cancer diagnosis to death, but more sophisticated examples might be something like the lifetime of a battery or mechanical component of a piece of machinery, and maybe even more new, something like the time from a user going onto a website and then clicking on an ad, which would be relevant to something like A/B testing. One of the main things that survival modeling accommodates is this idea of censoring. So censoring is what happens if we don't see this event before the study period ends. So if we're watching cancer patients, maybe we only watch them for 5 years, and all kinds of things could happen in those 5 years before they get cancer. They could have a heart attack or a stroke, or maybe they live to those 5 years and we don't see the event. So all those people would be censored. And so that's a type of missing information where we don't know what would have happened if we had seen them indefinitely. So we don't know how long it would be before they would get cancer theoretically. So this is an important type of missing information, and this is something that survival models are designed to accommodate. So just an example, like I had previously mentioned, an example of this would be observation of a clinical trials patients for 10 years, and we want to see if cancer has recurred, and if they're censored if they die before cancer recurrence, or maybe if they last beyond that 10 years and don't get cancer. So the goals of this project that we've been working on, there's 3 things with survival analysis that become really important for us to, that we're interested in, that we want to quantify. So one is the survival curve. So this is the probability of the event happening over time. So at time 0, nominally, we have 100% of everything is working. And then you can look out in future times and see how those probabilities will change over the course of a study. Something else that can become very interesting is something called the hazard rate, and you can think of this as maybe the immediate risk of an event occurring. And this one is a little trickier because it's difficult to estimate mathematically, so it becomes not as straightforward as some of the other estimates we're talking about, like the survival curve. And then lastly, we of course want to know how different covariates and measurements might affect the time to of survival. So for example, we can think of cancer patients. Maybe they would have some kind of treatment that we think prolongs how long it is until cancer recurs or something like that. So these 3 things are something we're very interested in. Today, one of the things I'm really going to focus on is this method for estimating of the hazard rate. And I'm just going to show a little graph here on why the hazard rate is so interesting and important. So on the left here, this is sort of a hypothetical situation where we have a hazard rate, and we can see that this immediate risk of failure actually increases over the first, say, 2 years of a study, and then decreases over time. But if we look at the survival curve, which is a more aggregate measure, which is on the right, we really can't see that initial hump of risk, that sort of gets washed out in the measurement. So this just shows an example of why we would even care about the hazard rate and what additional kind of information it can provide, and why we would bother to estimate it, even though it's trickier than some of the other functions. One of the things that we have been working on in the past couple years is addressing some common statistical complications that are occurring more and more in these types of data. So storage has become easy. We have all of these methods for retaining more information about different patients or objects. And on top of that, medical treatment is getting a lot better. So we have these issues with sparsely observed failures. So for example, you can think of some cancers are very, very long-term. People can live a very long time with these, with different types of cancers. So we may not see a bunch of people getting cancer. And because we don't see a bunch of people getting cancer later on in a study, that can make some of the common methods for estimation fall apart, or those estimates degrade a bit. And so one of the things we focused on is how can you estimate a hazard rate even when maybe we don't have that much data to observe in the end of a study. The other thing that becomes important is the fact that the effects of covariates can change over time. So the most common assumption in survival modeling is that the effect of treatment, for example, will have the same effect over the course of an entire study. But if we think about this intuitively, it might not make that much sense to think that a treatment could have the same effect in the first 2 years that it would 10 years after it's been given. So we're going to examine both of these things today from a statistical modeling perspective. The data that we're going to look at today, this case study or example, is prostate cancer. And so prostate cancer is a really classic example of cancer that is common. It's 26% of the cancers among all men, but people can live a really, really long time with prostate cancer, and it almost can be thought of as a chronic disease. So not that many people die of prostate cancer. So when you're looking at best kinds of treatment, you can have men who live for a very, very long time, which both contributes to both of these issues where we don't maybe see that many failures at the end of a study, and where the treatment effects might change over time. The data that we have specifically is some data from some clinical trials, RTOG clinical trials, and what we're particularly going to focus on today is the time to prostate cancer recurrence after a treatment. And this is called biochemical failure in the clinical world. But you can think of this as prostate cancer recurrence. And the main thing we want to focus on, we have a bunch of different things we're interested in, but the main thing that we want to focus on is 2 treatment groups. One is short-term treatment and one is long-term treatment. And the basic idea is that we know that long-term treatment will reduce the risk of cancer coming back, but it also has a lot of side effects. So being able to quantify exactly how long it will take before the prostate cancer returns, and if there's a real benefit from this treatment is really important for the people who are actually getting treatment for prostate cancer. So this is the first issue I brought up previously with these sparsely observed failures. So if we look at the number of, this is just these little 2-month bins, and if we have a 12 years worth of data. So if we look over this period of time and we look at how many men have cancer recurrence, we can see that in the very beginning on this graph on the left, that there's a lot of failures in the beginning. 30 or 25 over these 2-month periods, which then goes down all the way down to 0. So So near the end of this study, we really, past like 6 years, we really don't have that much information because not that many men are still having cancer recurrence. And the opposite of that, when we were talking about censoring and this concept of missing data, we can see that towards the end of the study, this is the graph on the right, we can see that the number of men who are censored increases over time. So we're seeing fewer men who actually have cancer recurrence and more men who are censored for one reason or another. Could be, which makes sense, it could be a stroke or a heart attack or some other thing that's not prostate cancer that removes them from the study. Some of them even go all the way out to the end of the observation period. And so we don't know whether or not they would have gotten prostate cancer. The second issue that I had mentioned was the fact that the effects of some covariates like treatment can change over time. So this is sort of a hypothetical situation here where if we use the standard assumption that these treatment effects are the same across the course of a study, we could see that we have two hazard rates here, one for short-term and one for long-term treatment, and that's on the left. And these would be proportional to each other. And on the right, we see what happens if we take the log ratio of those two hazards. So if we use the standard modeling assumption that this treatment effect is constant, this is called a proportional hazards, then it wouldn't change over the course of the study. But the reality of what this data looks like is more like this. So we can see that, first of all, on the left, these hazard rates definitely don't look like they have the same shape. And on the right, if we look at the log ratio of the two hazards, we definitely see some kind of changing effect. So this is actually called, we can call this non-proportional hazards. So for people who are not familiar with survival analysis, this is sort of a way of discussing a effective treatment or some other measurement that changes over the course of a study. So these two things, both the sparse failures and the changing effects of covariates over time, really can make the mathematical modeling of time to event data tricky. One approach that I've been working on with Vanya on this on my postdoc is this multi-resolution hazard model. So this is where I'm going to just kind of talk about some statistical details of this model. And then after this, I'm going to talk about how it can be implemented in the accompanying R package. So this model, for people who are familiar with Bayesian modeling, this is a Bayesian model. It's semi-parametric, and it will produce an estimate of the hazard rate and the effects of any covariates on the survival times. And the prior for this model is a type of Polya tree. So a Polya tree is some kind of measurable space that gets recursively split. And with the MRH model, the number of times this tree is recursively split into two is fixed. And we can then estimate the hazard level at each level of this tree. And I'm going to show some graphics on this so that to make things a little more clear. So since this is a Bayesian model, it has this prior tree. This is the type of Polya tree that I was talking about on the previous slide. And our measurable space is the cumulative hazard H, which is at the top of this tree here. And this is an example of a Polya tree that is split into 3 levels. So what we're going to do is we're going to estimate the hazard rate on the bottom levels of this tree. So we split this cumulative hazard over the course of the study with these split parameters, which are these R10, R20, etc. And those will control the height of the hazard rate. And we estimate the values of these split parameters with the data. And then these Ds at the bottom, these are hazard increments within each bin. So there's 8 estimates here across the course of the study. And we can write these as a function of our cumulative hazard H and the split parameters. So what does that mean? Well, if you're looking at, let's say that this yellow curve is a hypothetical hazard rate, and we're going to look at how splitting this tree can estimate the hazard rate. So in this first upper left graph, we see that we just have our top level of the tree. It's just a flat line. But if we look at this second graph on the upper right, that's our first split parameter, and that'll be estimated using the data, and it's going to be greater than 0.5 because the first half of that hazard rate is higher than the second half. Similarly, if we look at the lower left graph, we have two more split parameters. That's the second level of our, or the second level of our tree. And these are also both going to be greater than 0.5 because in the first half of the study, the hazard rate is higher. And in the second half of the study, this first increment is higher. And then lastly, if we look at the lower right portion, we can see that we would have a similar pattern. This first split parameter would be less than 0.5 because it's that first part of that split is smaller than the second half. Ideally, you'd have something like this in the end where you've done the tree to a fine grain enough level that you'd be capturing most of the details in the hazard rate. Again, because this is a Bayesian model, we use MCMC sampling to estimate parameters. And I'm not showing the likelihood function here, but it can be written as a piecewise exponential model, which is convenient for notational purposes. It has the strength of the tree behind it, so that gives it some advantage in certain scenarios when compared to the piecewise exponential model, but they can be written in the same manner. And then we put priors and hyperparameters and hyperpriors on the parameters of the tree. And when we have some non-proportional effects, so those are the time-varying effects, we have separate strata for those. We have another tool that we employ with this model, which is the pruning. It's a pruning tool, and I'm not going to go into too many details here, but this is a technique that lets us merge two bins if it seems like the hazard increments are statistically similar. And we have a paper on that if you're curious to read more about that. And there's some, it's data driven. It can reduce the amount of computing time because it reduces the number of parameters that need to be sampled. And it also, because the number of parameters that are sampled is smaller, we can also produce robust estimation even when we're not seeing, for example, that many men failing from prostate cancer in the second half of the study. Okay, so back to this application where we're looking at prostate cancer recurrence, and this is the clinical trials data set, and we have over 1,400 men and they're followed for over 13 years. And today I'm going to do an example on how we would analyze this data if we have 4 levels to our tree. Ideally, we'd have more for that, but for the purposes of time, we're just going to use 4 levels of our tree today. And then we include treatment, letting the effects of treatment vary over time. All of this can be implemented in the MRH package. So some of the actual modeling can be tricky to code, so this makes it convenient to actually use this theoretical tool. And so we have the MRH model, the covariates can be made non-proportional. The output includes chains for the people who are familiar with Bayesian modeling, diagnostic graphics, and point estimates. And it also conforms to the standard R software development practices. So for anyone who's familiar with modeling in R and survival modeling in R, this stuff will look really familiar and not require a bunch of extra learning to figure out how to use. So now I'm going to show how we would run this software in Domino. All right, so I'm going to demonstrate how the MRH library and package will work in Domino. So first thing I'm going to do is I'm just going to open an RStudio notebook so we can look at some code and see how this package works. Okay, so first I'm just going to show, first we just need to install this package onto Domino's, so I'm going to do that first. It's called the MRH package, so pretty straightforward there. And then I'm going to load in this data. We can look at the first 5 rows if we want. All right, we have the time to biochemical failure or cancer, prostate cancer recurrence. We have whether or not they were censored, so whether or not we actually saw them have biochemical failure if something else happened first. We know whether they were in the treatment group, the short-term or long-term treatment group. And then these last predictors are important. They're just measurements that kind of let us know how severe the cancer is. And I'm not going to go into detail about those, but those are other important things clinicians are interested in. So the main function we want to look at is estimate MRH. So there's a lot of information here for people who want to know more, but the main thing to kind of know is that we're going to enter a formula and the data, and then we need to enter how many levels of the tree we want. So I'm just going to make a model here first. Some important code. So here's our estimate MRH. This is the standard survival formula that's used by R. Right now, this only does right censored data, so it won't accommodate interval or left censored at the moment. And then we have all our covariates in here. This is the standard. These are all following standard R practices. And then this NPH here, this is letting the function know that treatment should be included so that it is allowed to vary over time. We give it our data, we're telling it to put 4 bins, and the maximum number of iterations is going to be 1,000, which this is not nearly enough. We should probably do more, but for the sake of time and resources, right now we're just going to show this at 1,000 for this particular example. So while that's running, it's going to take a couple minutes to run. I'm just going to show here, I had specified, since this is a Bayesian model and it's doing Gibbs sampling, you can change the number of iterations that you want. Default is 1 million. It usually does not need that much. But you can change that. That's flexible. You can also change the thinning value. So the default value is 10, but this can be changed to whatever seems reasonable for reducing the autocorrelation. And then you can also change the burn-in period. The default is 50,000 unless you've picked some smaller number for maximum number of iterations. So those are ways in which you can control the properties of what will be retained in the chain. You can also do some pick your initialized values, change some of the prior parameters and hyperprior parameters. And there are some other options here for checking if the chain has converged. And so those things, I'm not going to go over in detail, but those are options for users who want to adjust. And of course, if you don't want to adjust any of those things, then you can just let it run on its default mode. So now we see that this has finished running. We get this warning message because, like I said before, we didn't do enough iterations, but for the sake of example, I just wanted to shorten how long this would take to run. And so we can do this model.full has a lot of different parts to it. We can see the summary contains the example, contains the parameter, the median of the posteriors, the marginal posteriors, and the quantiles. And then we can see the hazard rate, the effects of the covariates, and all kinds of other things that we might want to know about this particular model. The MRH package also includes the S3 functions. So, for example, we can just use the plotting function right here and kind of see it's a little hard to tell what's going on. Group 0 and group 1, it's a little hard to tell. Those are the solid lines are the medians of the distribution of the hazard rate within each of these 16 bins. And the dashed lines are the quantile bounds. So it's a little hard to tell. So one thing we can do is use this and say combined.graphs equals false. And then we get two separate graphs here. It's a little easier to tell. This is 0 group is the short-term group and 1 is the long-term group. So we can kind of see how the two hazard rates compare. We can also see the if we look at model.full $beta, these are the actual, these first few are the effects of the other covariates. And then we have these ones here. And then these last 16 are the log ratio of the treatment effect over time. All right, so now I'm going to stop this studio session. I'll rename this. And what I want to do is I want to compare this to the, I'm going to view my results first of all. So one thing, besides the graphs that we made, the MRH package also provides a number of files in a folder to the user. So here, for example, are some of the, and it's titled MRH results. And so here are the different hazard rates. It shows you the baseline hazard rate and a smoothed version of it. It compares the two groups. So there's a bunch of different graphs. It also provides the chains. So this is the value of the parameter at each of those iterations that's retained. This is the sampled value. So you can look at the whole posterior if you want to. It shows us some more figures, and we also get some diagnostic graphs. So this is a way for us to assess whether or not the chains have converged and are running properly, etc. These don't look that great because we only ran it for 1,000 iterations, but hopefully these would look, there wouldn't be any autocorrelation, and your trace plots wouldn't have any trends in them, etc. So this is also something that's automatically provided to the user so that you can do a quick check to see if everything looks generally okay with your sampling. So now what I want to do is I want to compare these to some other models where I include that pruning tool I discussed, which kind of reduces a number of parameters and can make the sampling routine run faster. So I'm going to run those. I'm not going to run those in RStudio. I'm actually just going to run those as their own files, and then I'll show you what's contained in those files while that is running. So this is going to be similar to what we did before, but this one is going to be the one with two prunes, 2 levels pruned, and this is going to be with all levels pruned. So while those are running, I'm going to show what that looks like. So here are all my files, and let's, for example, look at what this code looks like when I want to prune 2 levels. So this is all the same as before. And actually, this is all the same as before as well. So nothing has changed yet compared to what I just did. But now I'm telling it to prune the model. So I'm telling it to prune the prior tree, combine similar bins if there are any. And this tree has 4 levels, so I'm telling it to only prune the bottom 2 levels. And I don't want to overwrite my previous results. So instead of my default out folder, I'm going to put this in a different out folder than before. And similarly, when I'm pruning all levels of the tree, I have a similar thing, except for now I'm telling it to prune all the levels of the tree, and I'm giving it a different out folder. So most of this code is the same, except for this very last line. Now we can go to the results of this pruned model. So just like before, I can kind of assess how these what the hazard rate might look like if they're pruned. We have a little less detail here than in the previous one because, for example, these bins are fused. And I still have my chains and all of my diagnostic graphs and everything else I might want. So I have those for both this prune all and prune 2. So, so now that we see the code, the last thing I want to do is I want to show how these models compared to each other, how can we assess them? I'm going to do that again in RStudio. This is going to be a post-processing session. And you'll notice that down here I have all my results, so that makes it really convenient for me to, even though these were submitted in the background, I still have them here, which makes it really convenient for me to look at them here. So the MRH package, I'm going to reinstall this because I actually don't need this. The MRH package has some functions that really help in the post-processing of the data. And so if you'll remember in the results, one of the things you have are these chains. So let me show this again here. Oops. It's not gonna work. So one of the things that's given to you as a user is this file of chains. So there are some post-processing functions in R that, let's say you run all these on in the background or submit them or schedule them in a queue for later. You can still go back and have all the information you need. You don't have to do this on an open console. So what I'm going to do here with these lines of code is I'm going to read in those chains and I'm going to make them an MRH object, which then allows me to use some of these MRH package functions to process those chains conveniently. So now this still looks the same. This still looks like a chain. Looks like the chains. I still have all my chains in here, but it's now an MRH object. So now I can use this summary, for example. And now this summary function will automatically process those chains for me, so I don't have to calculate all these point estimates and quantiles on my own. I can also, the only thing that's different from the first initial code I showed is that you need to specify when the study ended. So this one ended at 160 months over 12 years. And I can also do a plot just like before. This is the same thing that we saw in the previous piece of initial code. Another thing I can do here, I'm going to plot side by side. I'm going to plot my hazard rate, but I'm also going to plot my survival curves. So there's a plotting option for here on the left survival hazard rate and on the right survival curve, which some of these are hard to read, so you can decide if you want to separate those. But another thing we want to do, since we've made 3 models here, one has no pruning, one has the bottom 2 levels of a tree pruned, and one has all of the levels of the tree pruned, is using this DIC function. And so for people who are familiar with model comparison metrics, DIC is deviance information criteria. It's used for Bayesian models. And it's used similarly to AIC or BIC. And so this function actually calculates an approximation of the DIC and also the AIC and BIC. And of course, with all these model comparison criteria that I've just discussed or just mentioned, the lower is better. So let's say that we want to compare these 3 models. One way we could do would be to calculate the DIC and AIC and BIC of each of them. And so we can see if we spend a little bit of time looking at these, that the model where all of the data is pruned actually has the lowest values. And the model where none of the levels of the tree are pruned has the highest values. So that would provide us evidence that this model where we've pruned all the levels of the tree might be the best fit to this particular data set. Another thing that we can do is plot how these models, how much these estimates change when we're comparing them. So how much these estimates change and how can we compare them. So I'm just going to do some plotting of the model summaries. So here, this first set of lines, I'm just going to store the summaries of these. So this has the hazard estimate of the hazard rate, which is the median of the marginal posterior in each bin, and the 95% credible intervals. And same thing for beta and the survival curve and the cumulative hazard and a few other parameters that we might be interested in. And all I want to do here with this line of code is, these 3 lines of code, is I want to plot what the estimate of the log ratio of the treatment effect looks like when we have different levels of pruning. So that's this graph here on the left, and we can see that the estimates don't actually change that much when we prune them more. They're just some flatter periods. But one thing that does change a lot, what I'm going to plot now are the credible intervals. There is a bigger difference here. And so you can see here that these intervals, let's put a little legend on this, that when we don't do any pruning, those bounds are definitely the biggest. That's the black lines there. And then the red and the green ones are much narrower and smaller. And so that makes sense because there are fewer parameters there. And the green and the red ones are the 2 levels, and all levels pruned are very similar because there aren't that many parameters that are actually pruned in the upper level of the tree. So it's sort of a raw way of comparing the different models. So now what I want to do with this graph is I'm just going to export it. I'm going to just save it to my file here. So I have my files. There's my rplot. I'm going to sync this so that I'm sure that I have it in my files here, and it's right here. And obviously I can make that more pretty if I want to spend a little bit of time working on the specs of that plot size, but there it is. So I can sync it that way. The other way that it would sync is automatically if I am in here. Let's say I want to do this again, and I want to do our plot 2, save. Now when I stop running this, I also have our plot 2 in here. So there's 2 ways to sync that, both of which can depend on how exactly the information is being shared. So that's how that would be run in Domino. One of the main questions I usually get is why there's a lot of survival models out there. How you know, how does this one perform? And so we have a paper on the archive actually that you can check out if you want. This is sort of a brief graph of that. What we did was we simulated some data and did some comparison between the MRH package and a variety of other survival packages in R. And these are some of the results we see, kind of just assessing how well these packages perform in a simulated setting, and some of them perform better than others, so you can kind of check that out, but some interesting stuff there. And yeah, so I have a number of collaborators I've been working on, but obviously my postdoc advisor, Vanya Dukic, and then Jim Dignam, who works with the clinical trials data, this prostate cancer clinical trials data, and Yuangting Chen, who was a grad student while I was there during my postdoc. Thank you.